Dear audience, today's topic is the plurigenomics or multigenomics. These two words was coined by me, but you will understand that this is really the applicable common notion. The study plan includes the introduction, the basic notions, the availability of external compounds, the definition of the plurigenomics, the process of protein synthesis, the regulation of gene expression, where are the attacking points of the external and maybe internal chemical compounds, the omic technology basics, what is a microarray or a gene chip, and the absolutely new field of the metagenomics will be shortly presented. And finally, the veterinary and medical human application will be given in short. First, we have to see which are the factors which influences the metabolic state of a human. The or an animal. It is influenced by internal and external factors. The internal factors are the body composition, more or less fat percentage, etc., the tissue metabolism, the level of basal metabolism, which is in close relationship with the thyroid hormone levels and activity. Age, of course, generally the genotype, the health status. These are the background and bottom, and the external factors may influence and may exert their influence through the major nutrients like the fat, carbohydrate, and protein from the diet. The active substances of the diet, like the minerals, the vitamins, the vitamin-like substances, etc. Similarly, the medicines or drugs may influence the metabolic state. Of course, the physical activity should also be mentioned. A very important role player is the microbiome and milliards of microbes in the gastrointestinal tract. And of course, the mental state has also some influence. Here we will see on this figure, you can see the causes and the dietary responsiveness of diseases. And you have a line, and the genetic end of the line shows that which is close to that point are mostly determined by the genetics, by the genetical background, like the progeria or Alzheimer's disease. The other end of the line are mostly influenced by the environmental factors, like the vitamin deficiencies. Here you can see folacin, or protein energy malnutrition, different types of cancer and liver cirrhosis. And in the middle, you have different diseases and pathological situations when both the genotype and the environment, like the feeding, drinking, and other, may cause the problem. like the anemia perniciosa, osteoporosis, ischemic heart disease, Parkinson disease, different types of diabetes, the hypertension and stroke, altogether the metabolic syndromes, the Wernicke-Korsakoff syndrome from the alcohol intake. So this is how we have to see the different metabolic problems and other problems. We have to add that some independently, that is genetic or environmental, can be treated or influenced by the feeding and medicine, and other cannot. For example, the osteoporosis is partly genetic, but by the intake of the calcium and vitamin K and D, we can improve the situation. Or I have to mention the metabolic syndrome and the other as a medium, and the vitamin and mineral deficiency absolutely can be treated by the feeding and nutrition. What are the genes and the genome? Basically, the gene is a section of the DNA which codes, which may carry the code of one protein molecules, and these protein molecules may be a structural protein or maybe a functional protein like an enzyme. And this will give in the phenotype one characteristics. This is related to the sequence of the base on the DNA. What is the genome? The genome is the totality of the genes of an organism. Now we have to see the different names and sciences. Genetics deals with the individual's genes, like the Mendel rules. The genomics deals with the function of the whole genome. The epigenetics is the science of the changes of the epigenetic signals. And the plurigenetics will be given later. In the genetics, we will find mutations. In this case, there is a deletion or change of bases. In the epigenetics, this is a signal on the DNA and histone, like methylation and acetylation, and other also. The plurigenetics are changes in the gene functioning under the influence of external or internal factors. External factors are the feeding or the food intake, drugs, toxins from the environment, endocrine disruptors, irradiation, and even the social effect, and even the inner mental state. The availability is the keyword that a chemical may act or cannot act because only those chemicals can have an effect which can get into the cell. How can get chemical compound into the organism? By the air through the lungs. In this case, the determinant are the concentration and the particle size. Percutin, in this case, should be soluble in lipids or having chelated binding. The cosmetic industry developed a lot of methods to make otherwise no percutan soluble compound to go through by soluble in lipids and by giving in form of chelated by ETA, for example, EDTA, and for example, vitamin C can be made possible to go through the skin. And peroral, this is the main topic, and you know that the first member of this orchestra are the non-members of the microbiome, and they can change the dietary intake and also can synthesize new molecules. But this is only a precursor for the liver metabolism. And after the liver metabolism, what is in the bloodstream may enter into the cell. Of course, in case if the internal background, for example, for the sugar, the insulin is present, interactions in the lumen of the gastrointestinal tract may influence the availability of chemicals. A very well-known example that by calcium overeating, one can decrease the availability and the absorption of the zinc, and in this way, a secondary zinc deficiency syndrome can occur. It was an interesting case 30 years ago when the limestone used to be very cheap compared to the other component of the mixed feed for fattening pig, and they put more limestone in the mixture. The consequence used to be a parakeratosis in fattening pigs owing to secondary zinc deficiency. And of course, for a medical person, the state of absorptive surfaces, for example, inflammation, parasitic presence of parasites, etc., may change the absorption rate. But if a chemical compound get into the cell, is capable to exert an effect, and this effect may have different lengths and different duration. If this external effect used to be a strong, like chemicals on the fertilized egg or irradiation, may cause mutation, may cause deletion, which will destroy the cell or the organism. If not, it can be transmitted to the next generation. A moderate effect may cause epigenetic changes on the DNA and histone signaling signals, etc. These are measurable changes, but the bases, the nucleotides, did not change. The constancy is limited, and the transgenerational transmission is limited if the effect is during the formation of the germ cell lines, the sperms and the eggs. And finally, the weak so-called plurigenomic effect may change the gene functioning, the gene expression, but no traces like previously. The environment may modify everything, but the efficacy, the power and duration is different. Here you can see the first example for the strong effect. Cause of sickle cell anemia during the evolution used to be only a change in one nucleotide, namely instead of in the normal hemoglobin, you had glutamic acid number 6 and in the protein, because in the DNA you have the adenine, which changed to thymine, and number 6 amino acid changed in the sickle cell anemic person in valine. And this is the only genetic change, and they survived. Even they have some lower, they have some resistance, it means lower sensitivity to malaria. Okay, example for the epigenetics changes, it may be a DNA methylation or demethylation, a histone acetylation or deacetylation, the rearrangement of the chromatin before the cell division, the microRNA effects, the so-called sumoylation and ubiquination, and one can continue the series, but it's enough for today. And lastly, the example for the plurigenomics, which is a change of the gene functioning, the gene expression under the influence of external, and now we can add internal factors through a chemical compound. This is the key, that there is a chemical compound, external or produced internal. No changes in DNA base sequences and no change in epigenetic signs described previously. So you see that the plurigenomics, similarly to the genomics, measures the changes in gene expression throughout the whole genome simultaneously. And according to the origin of the compound which exerts this effect, we can have nutrigenomics when the compounds of the diet are the players. We may have a pharmacogenomics and toxicogenomics when medicines and toxins are the chemicals in the center. But we can have also eto or psychogenomics when in the change of the behavior will modify the hormone levels, for example, in the body as chemical compounds. Or last but not least, the radiogenomics or irradiogenomics, when owing to a irradiation, new chemicals are produced in the organism, which is the signal for the changes in the gene functioning. These factors are acting through an external and internal chemical compound. Therefore, we can give the common name that multi or plurigenomics. How is in details this gene functioning? The stages in the gene expression is similar in the whole living world, but here we will see the eukaryotic cells, which we have also nucleus. In the left side you have the events inside the nucleus. You see that from the DNA, after the signal, from the DNA, the gene available for transcription will transcripted to messenger RNA. But this messenger RNA should undergo to a splicing to a maturation, and after we will have the real messenger RNA, which will, and see the right side of the picture, this is in outside the nucleus, inside the cytoplasma. The messenger RNA may survive or may be degraded, but until surviving time will give a form for polypeptide and protein synthesis. The building stones for that are transported by the transfer RNA, and the site of the synthesis are the ribosomes. Yes, and please look inside the first step, signal, chromatin, and DNA. And you see that in the DNA we have promoters of the eukaryotic gene, which is the starting point of the messenger RNA copying, and you see that this promoter binded with transcription factors, and if binded with transcription factors, the so-called RNA polymerase enzyme start the transcription. And in this event, the transcription factors has some receptors and can receive the outer chemical compound, which may change, may be enhancer, activators, and maybe inhibitors. Here you see that we have a network of transcription factors. It is only an example, which are through the ligands of their receptors are fixed to the promoters. And these ligands may be also some chemicals from outer world. You see here, for example, the later explained PPAR, which is a peroxisome proliferator, activated receptors, etc. All these are highly important in binding the outer chemicals, the vitamin A, the vitamin D, the fatty acids, and the series of hormones, etc. And where can this gene expression, the first step, regulated? It can be regulated at the first step, it is the transcription. But it can be regulated during the translation. This is the last step from the messenger RNA protein. But the messenger RNA processing, when the maturation inside the nucleus is going on, this is also can be sensitive to other compounds. And the messenger RNA stability in the cytoplasma, or the half-life time, will determine how many products can be received from this messenger RNA. And the basic of the ribosome and the transfer RNAs are also important and can be influenced. Okay, back to this so-called chemical compound of anything, because this is the general device, because the behavior is also caused by chemicals. you have to see the neurotransmitters, the cortisol in the brain, and the radiation. Irradiation will change the chemical milieu interior too. So after that we speak about the chemical signal. This may be number one, a ligand for transcription factor receptors. For this, a good example are the fatty acids, the eicosanoids. They bind to PPAR, peroxisome proliferator activated receptor. Number two possibility, after having metabolized the chemical compound, they will change concentration of substrate and intermediates in the cytoplasma. And of course the pH, etc. For that, an example is the cholesterol sterol metabolism in the cell, for example in the liver cells. Number three possibility, that the chemical compound may influence the signaling pathways. For that, a good effect, how the green tea's polyphenols may exert the anti-cancer effect. So the PPAR ligands, a little bit of illustration, may be the fatty acids of the food, and the eicosanoids, the omega-3 and omega-6 fatty acids of the food. They are natural ligands for the PPARs. We have synthesized more potent synthetic PPAR ligands, including thiazolidinediones and fibrates. They are effective in the treatment of diabetes and dyslipidemia of humans. This pioglitazone is a thiazolidinedoine insulin sensitizing drug whose effects results predominantly from its modulation of the transcriptional activity of the mentioned peroxisome proliferator activated receptor, in this case gamma. Alpha, beta, and gamma PPAR are the most important. How to measure the gene expression? We can measure in vitro the gene expression using microarray or gene chip. These are collection of microscopic DNA spots attached to a solid surface. It is for measuring the expression level of large numbers of genes simultaneously. Each DNA spot contains picomoles of a specific DNA sequence. And this is working with colors, active, expressing, or inactive, but you can have other red and green colors, and previously, of course, they determine that which point which gene can be. And we have to see an extremely new approach in the frame of the genomics, and this is the metagenomics. The metagenomics means that the genes, DNA collection are sequenced. Where this collection come from? For example, from the microbiome, from the feces, or from the environment, and is analyzed as a single genome, this sample. Isolation and laboratory cultivation of the individual microbe species like bacteria, viruses, bacteriophage, fungi is omitted. Genomes are studied in context with the environment, with its and their environment. And the resolution of the study are not at the level of the species, but at the level of species population. Here is a summarizing picture from the Wiki Educator. You see that up sampling from the environment or from the feces or from the microbiome after some extraction and manipulation, we can make a function-driven analysis to see the most important processes, or a sequence-driven analysis. For that, we have libraries, DNA libraries. For further reading in this topic, I can propose a chapter in Koch's book, Introduction to Marine Genomics. The authors are Meyer-Dirks and Glockner. And important news that the same chemical compound may have epigenetic effect and plurigenomic effect. Here you see the whole process from the chromosomes through the DNA, and Me stand for the methyl group and methylation. And an extremely good example for this pluripotent pleiotropic effect, that the Nutri and epigenomic anti-tumor effect. Garlic's allyl mercaptanes, the sulfur-containing compound, inhibits histone deacetylase and stimulate at the same time binding of a transcription factor to a promoter. And all these leads, at least in vitro, but hopefully in vivo, to the apoptosis of liver tumor cells. Nia et al. 2008. What are the possible medical, veterinary and human medical applications? First of all, go back to the fats, oil and specific fatty acids. They are regulating factors, transcription factors and genes. How? The peroxisome proliferator activated receptors, the alpha, beta and gamma, are distributed in the different tissues and differently. And this is why, for example, the different fat tissues in the different locations in the stomach or in the bottom have different metabolic activity in fat burning and in causing obesity and metabolic syndrome. The number two, the fatty acid synthase gene is also regulated by the fat, oil and specific fatty acids, and the function is the fat synthesis, which will stimulate the fat synthesis and the background gene expressions. The fasting, for example, the fasting and the different slimming diets activate lipolysis, and these is at the same time an increase in the free fatty acids in the blood. It's similar as eating too much fat and oil. But be careful, on the contrary, the refeeding after fasting by fat-free, high carbohydrate diet will activate the fat synthesis by enhancing the fatty acid synthase transcription rate. And everybody knows that in the human slimming diet history as a yo-yo effect, or in the slimming diet of our pets and companion animals. What about the protein-rich diet? The protein-rich diet, in other words, low-carb diets at the same time, causes microRNA shortage for fatty acid synthase in the adipocytes, moderating total body fat. But no, unfortunately, in liver tissue. Therefore, protein-rich diet is good for the slimming diet, but maybe one can receive a fatty liver. What about the Atkins diet, which is a low-carb and high-fat, high-protein diet, and Duncan diet, high-protein diet? These are the the disadvantages of this diet. But there is a good news that fortunately the hepatic fat synthesis can be inhibited by polyunsaturated fatty acid intake, like omega-3 fatty acids in the diet. As mentioned, the dietary carbohydrates stimulates the transcription of fatty acid synthase gene, more fatty acid synthase gene, more fatty acid production everywhere, but in the liver, of course, and this is the background of the production of the foie gras, which is discussed by the animal protectionists. But this is depend also upon the method used. In human, the dietary carbohydrates can be limited in case of a low carb diet or ketogenic diet or anti-cancerogenic diets. And in this case, the fat production enhanced by the fatty acid synthase gene, enhanced by the dietary carbohydrates, can be eliminated. Okay, so low carb diet, ketogenic diet, and anti-cancerogenic diet. What about the conjugated linoleic acids, which are a very typical metabolically active group of the linoleic acids? The conjugated linoleic acids are potent activators of the PPAR, peroxisome proliferator activated receptors. And in this way, the conjugated linoleic acids found in the butter and found in the beef meat can induce apoptosis in adipocytes, decreasing the total body fat. Inhibition of pro-inflammatory cytokines messenger RNA is also typical for the conjugated linoleic acids, and by this way they have anti-inflammatory effect. But now you know the background. Especially from the conjugated linoleic acid, the best being the cis9 trans-11 conjugated linoleic acid or romanic acid, which gives more than 2/3 of the milk fat. Using rat model, the anti-carcinogenic effect was strong. They reduced the incidence of breast cancer. What about some metals? For example, the peroral zinc intake, zinc salt, of course, mainly, may enhance transcription rate in a metallothionein gene. Metallothioneins are small molecules, polypeptide molecules, which which have a sulfur content, and they are able to bind the free metals, the zinc and the copper and others, even toxic, in the blood circulation, decreasing the danger of toxicosis. Zinc also inhibits the gene expression of pro-inflammatory enzymes, COX-2. So in this case, this is thick oxygenase 2. In this case, the zinc react as an anti-inflammatory, like a non-steroid anti-inflammatory drug. In the practical field of the pet food industry, we have for the dogs G-Pro-D named veterinary prescription diet. G stand for joint, because they are against the arthritis and joint problems of the dog and cat, and they contain either eicosapentaenic acid or docosahexaenic acid in case of the cat. Both have anti-inflammatory effect based on the Nutrigenomics. What about the vitamins? The vitamin A, among others, stimulates the expression of leptin gene. And more leptin expression, more leptin production will decrease the appetite. And this is the reason that in canine weight reduction diet, we have increased vitamin A concentration, say from the normal 2000 international unit per kilogram, we can apply 3000 international unit. But be careful, because there is a danger of vitamin A toxicosis, so too high is not possible. What about the the water-soluble vitamins? For example, the biotin is necessary in the ornithine transcarboxylase gene expression. This enzyme, the ornithine transcarboxylase, is important in the ammonia elimination. In the supplementary treatment of high blood ammonia and hepatic syndrome, is beneficial to give more biotin per oral. This contains also the metabolic prescription diet, and in the human field, it is a possible constituent of the patient of cancer disease. The Epi and Plurigenomics and the Nutrigenomics and all these topics are related to the systems biology. Why? Because the warm-blooded organisms, human, animals, and the microbes make together a super organism. This is a absolutely complex ecosystem where the metabolic compartmentalization is typical. Uh, uh, Everywhere you have membranes and membranes and membranes, and differences in concentration. What are the influencing factors? The mentioned endogenous and exogenous. Endogenous, genotype, circadian rhythm, age; exogenous, food, feed, drink, xenobiotics; and intermediate, the microbiome, are determining health and, in case of animals, the productivity. And all these can be studied only with the system biology approach. And in the system biology approach, the metagenomics may have a very important role. Thank you very much for your precious attention.